It's up to you, but maybe it might be better to
At
have
the
a
end.
question at the end.
At the end,
Yeah.
yes, I agree.
And because
I
at
agree
least
with
you
that.
have an overview of the topic and yeah.
Correct. Okay, whenever you want, Kri.
Okay, so, well, welcome everybody. Today we have the great pleasure to have with us Shinji Deguchi, Osaka University. Shinji did his undergraduate studies in aerospace engineering at Nagoya University, and then he moved to Tohoku University for his PhD. He's got his first position as assistant professor in the Department of Mechanical Engineering in Okoyama University, then associate professor at Tohoku University and Nagoya Institute of Technology. And since 2016, he is full professor in the Division of Bioengineering of Osaka University, where he's leading the lab Fine Mechanics in Life. I actually had been following Shinji's scientific work through his numerous publications in mechanobiology field since quite some time. And then I finally had the pleasure to get to know him at the Japanese Molecular Biology meeting last December. And today I'm really excited to hearing his talk about his recent work on new machine learning-based approach traction force microscopy to predict cell mechanics from images. So Shinji, thanks a lot for accepting our invitation, and the floor is yours.
Thank you for the invitation, and so thank you for the kind introduction. So it's my great pleasure to have this opportunity. So the topic of my talk today is on the development of technologies that must be useful in mechanobiology. But I assume that you are not familiar with mechanobiology. So let me start from this cartoon. So this is a cell, and it is known that forces, physical forces, are generated within the cells. So by the interaction between myosin and actin in most of the cell types, including endothelial cells shown here. And it is known that forces affect many functions of the cells that are related to growth, differentiation, and apoptosis. And they are also known to be involved in many diseases, including cancer. So it's really important to understand the effect and role of mechanical forces in the function of the cells. But importantly, the force is not a material, so it's actually hard to identify the presence of the cellular force. So unlike the protein, for example, so in the case of proteins, you can visualize the presence of the proteins by using, for example, fluorescent proteins like GFP, or by using antibodies for immunostaining. But in the case of force, it's not a material, so it's not straightforward to observe the presence of the force. So, but because we really want to know the role of the force, so we somehow have to visualize the presence of the force. And another important point is that forces are regulated downstream of diverse signaling pathways. So it means that if you have some proteins that you are interested in, and if you apply some perturbations to the protein, because forces are regulated in such a complicated manner, so it's hard to predict how the force is going to change upon the perturbations to the cells or perturbation to the specific molecules. So it is actually very difficult to identify the presence of the force, but actually there are some techniques to evaluate the cellular attraction forces. And I think this is one of the most popular ones and reliable ones. So this is called traction force microscopy. So to do this experiment, microbeads, so these are microbeads, fluorescently labeled microbeads. So these microbeads are embedded within the gels, and by tracking the positions of the individual microbeads, we can evaluate the deformation of the substrate. And by doing so, we can also measure how much force is generated by the cells that are created on the top of that. So this fraction force microscopy is really good in terms of the accuracy of the measurement. But on the other hand, from my own experience using this traction force microscopy, by the way, traction force microscopy has been still developing by combining, for example, super resolution microscopy to increase the spatial resolution. But I think this has some limitations, and one of the biggest limitations that would be the lack of the throughput. I mean, so for example, when you do this experiment, we actually have to be very careful about the original positions of the individual microbeads. So otherwise, the measurement will fail to accurately measure the force field, because unless we get the original position or initial positions of the individual microbeads, then we would not be able to accurately determine the deformation of the gel and the force fields that cells are generating. So because we have to be very careful about the original position of the microbeads, so it will give some limitation in the throughput or effective or efficiency to get a large number of data. But as I mentioned, because forces are regulated downstream of diverse signaling pathways, so we really want to test a huge number of molecules to evaluate their effect on the force. But by using conventional traction force microscopy, because there's a limitation, so it would be hard to obtain such a big data. So we instead focused on this method. So this method has been reported by Harris. So actually these are cells, and they used silicon fluid. So this is a silicon fluid, and a part of the surface of the silicon fluid was treated with local heating. So by treating the silicon fluid by local heating, a part of the surface is polymerized or solidified, and by putting cell on top of that, when cells grab the surface and by making shrinkage, then wrinkle will appear. So these are wrinkle, and this is also a cell, and because of the contractile force, wrinkles are generated on the surface of the silicon fluid. So by the solidification, thin films are created, and by the buckling of the thin film, it shows wrinkles like this. And importantly, this is really easy to perform this experiment, because all you need is just putting cells on the solidified or silicon fluid. So, and it's also easy to confirm the presence of the force. So it is very good in terms of the throughput. But at the same time, on the other hand, it's hard to accurately quantify the force based on the information of these wrinkles. So even if we observe the wrinkles, it's actually hard to convert the information of the wrinkles into the actual force level or actual mechanical stress level. And another drawback of this method is that because the solidification is induced by local heating, so it means that the mechanical properties or the stiffness of the substrate may not be necessarily uniform, spatially uniform over the surface. So it means that it's harder to say which cells are generating larger forces, because the mechanical properties or the stiffness are different depending on the place. Okay, so we tried to overcome the difficulties that I mentioned. And so we modified the wrinkle assay that Harris originally reported. And we did that by using spatially uniform oxygen plasmas. And I'm actually skipping the details of the methodology, but I have published some of the methodologies in these papers. But basically, we use a spatially uniform oxygen plasma to allow the surface of a silicon elastomer to be stiffened. So once you put cells on top of that, so this is a magnified view of this cell. So as you see here, while cells are moving, you see that striped patterns appear and disappear in response to the movement of the cells. Okay, so
no representaba los reales, si bien
so actually these are wrinkles created on the surface of the substrate. Okay, so in the presence of the contractile force, wrinkles appear. But if you remove the cell, then the surface becomes plain again. So it occurs in a reversible manner. Okay, so this method can be used as an assay to visualize the presence of the cellular attraction forces. Okay, so these wrinkles are actually automatically extracted from the images using machine learning techniques. So green color shows the wrinkles automatically extracted from the images, and the red color shows the outline of the cells. So as you see here, wrinkles and the cells are extracted from the images, phase contrast images. And so these are actually non-muscle cells or cancer cells, but our system can also be applicable to muscle cells as well. So this is muscle cells. So actually this was obtained by collaboration with Professors Manabe and Fuji's group. And this elongated one is actually muscle cells. And you know, muscle cells respond to electric stimulation. So by giving cyclic electric stimulation, cells make a beating in response to the electric stimulation. And as you see here, wrinkles appear in response to the beating. So by observing the wrinkles, and based on the methods I'm going to talk later, we can convert the information of the wrinkles into the actual force or actual or mechanical stress that I'm going to talk about today. And this is another example that we used this wrinkle method together with micro patterning technique. So here's a micro patterned area to which cells can attach. So this is a single cell, and this is a time course of how cells get spread within this square micro patterned area with an area of 40 by 40 square micrometers. So while cells are getting spread, you can see that striped patterns or wrinkles appear. And when you look at the inside of the cells, actin fibers are observed. So even in your I hope even in your monitor you can see the individual actin filaments. I'm not sure about the if through this Zoom platform the resolution might be decreased, but actually actin filaments are running in this direction, and so blue color shows phase contrast images that shows wrinkles, and as this video shows, actin filaments and wrinkles are making right angles, so they are perpendicular to each other. So it's like if you pinch the skin of your hand, back of your hand, wrinkle will appear in the direction to which you are pinching or compressing. So similarly, if you got a cell with wrinkles running in this direction, it means that forces are generated in these directions. Okay, and this is another example that we put two cells, a pair of cells, within this micro patterned area with an area of 54 by 54 square micrometers. And while these two are interacting with each other, so as you see here in fluorescence microscopy, while they are changing the position and while they are changing the location of the actin filaments or stress fibers, as you see here, once again, blue color shows the phase contrast microscopy that shows the wrinkles, and while they are moving, actin filaments or stress fibers and wrinkles are always making right angle, so they are perpendicular to each other. So it means that wrinkles are generated by the contraction or shrinkage of individual actin filaments. Okay, so thus the wrinkles can be used as a readout of the presence of the cellular forces, but at the moment we are not still sure how much force is actually generated within the cells, even if we are able to observe the presence of the wrinkles. So to allow us to know the actual value of the force, we use the machine learning technique known as GAN, Generative Adversarial Network. And so to do this experiment, to allow this system to work as a detector of the force, we simultaneously performed two different experiments. One is wrinkle assay that I have mentioned. And by doing this, so actually there is cell here, and maybe you see that wrinkles are running in this direction, and these wrinkles can be extracted using this machine learning technique as shown here. And simultaneously, we performed the traction force microscopy that I mentioned at the beginning of my talk. So as I mentioned, to do this experiment, microbeads are fluorescently labeled microbeads are used. So these tiny dots shows the microbeads, and by analyzing the deformation of the substrate by tracking the individual microbeads, we can evaluate the force field or stress field as shown here. So what we did was that we performed these two experiments simultaneously, and using this GAN network. Okay, so using this system, we are now able to understand the relationship, the geometric relationship between this and this. So this network understands the geometric relationship between the wrinkle patterns and stress patterns. Okay, so this is it's like a translation of language. So now I'm speaking English, but if I got a smart translator, they would be able to translate what I'm talking about. Even if I talking in Japanese, for example, that language can be translated to other languages like English, Spanish, or whatever this professional translator knows the relationship or grammatical relationship. So similarly, this GAN system understands the way of translating between them. So, which means that if you got a new image of the cell like this, and once again, this is actually very easy to get this type of image. So all you need is just putting cells on the silicon substrate, and by putting the cells, our cell will exhibits the contraction, and based on that, wrinkle will appear as shown here. So this is easy to obtain, and if you got this type of image, the input of this image to this network provides a stress field that corresponds to this wrinkles. Okay, so like translation of languages, they are convertible. So because this is easy to get, so by getting a huge number of data, all the data can be converted to ones that containing the information of the stress field or force field. So actually, this is a collaborative study with. So he's my student. Hong Han Lee is my student, and Dr. Daiki Matsunaga, he's my colleague. So he's actually one of the corresponding authors of the manuscripts that we are now submitting to a journal. And so these are three representative examples. So this is example A, B, and C. And so once again, this is easy to get. And once you get this type of image, wrinkles can be extracted automatically, and these wrinkles can be converted to ones containing the information of stress or, in this case, displacement field. And these are actually videos. And if you pick up one, so white cells are moving. So here, purple color shows wrinkles, and wrinkles are now converted based on GAN system to stress field. So here, the arrows shows the direction. So here, this now I'm showing what I'm showing is the principal or stress. Okay, so that is a vector. It's like a force, and also this arrows shows the direction of the principal or stress, and the magnitude of the arrows or the color shows the magnitude of the principal or stress. So our system can easily visualize not only the present but also the actual magnitude and the direction of the local or mechanical stress that individual cells are generating. I think that this must be a new approach to predict the cell mechanics only from images. Okay, so but we succeeded in converting the wrinkle information into stress information just recently. That's what we are now submitting. So before 2020, we were not still successful in translating the information wrinkle into stress information. So before 2020, we used the length of the wrinkles as a readout of the cellular traction forces. So that's based on this experiment. So this is a cell, and to force the cells to generate wrinkles, we used, in this case, glass microneedle. So by forcibly displace this cell to this direction, wrinkles appear in response to this loading, and by removing the actually from the deflection of this needle, we can know the force, how much force is required to generate these forces. And by doing this experiment, by analyzing the relationship between the length of the wrinkles and the force required to generate these wrinkles, we can get this type of curve. And we found that there is a linear relation between the length of the wrinkles and the force. So based on this result, we tried to say that we try to evaluate the relative change in the force level before or after some perturbation are given to the cells. Okay, so but this experiment was actually justified based on our study using this machine learning system. So using this system, because this is a high throughput system, we found that indeed there is a linear relationship between the length of the wrinkles and the force that are required to generate these wrinkles. Actually, there is some threshold to generate wrinkles, so at least we have to give 10 Pascal or mechanical stress or principal stress, but above that, there is a linear relationship. So based on this result, we evaluated the relative change in the level of the force based on the length of the wrinkles that cells induce. Okay, so using this system, so at this moment, we were not still able to obtain the absolute value of the force or absolute value of the mechanical stress, but we are able to determine a relative change in the force level. And by using this system, we evaluated the effect of some focal adhesion associated molecules. So actually, this is another collaborative study with Dr. Ichikawa and Professor Kiyoka at Kyoto University. And also, at that time, we are really curious about how these three molecules that are associated with focal adhesions. So we are not sure how they are regulating the traction forces. Okay, so to determine the involvement of these three molecules on the in the attraction forces, we had to do a large number of experiments. So at least we had to test 24 different combinations to determine the effect of these molecules. And for each case, we had to do the experiment more than 20. So it means that we had to do the experiment nearly 500 different experiments. So, but based on our system, because our system is easy to get large data, so we finally obtained how they affect the cellular attraction forces to finally find statistically significant difference between so in the case of ARGBP2, this is one of the molecules. We found that this molecule is particularly responsible for an increase in the level of the cellular attraction forces. But unlike this protein, we found that other proteins are not involved in cellular attraction forces. Okay, and so far I have mentioned the application of our system to single cells, but our system is also applicable to many cells, multiple cells. So in this study, this is another collaboration with Professor Sakane and Professor Sasaki. So by using our system, we attempted to determine the effect of this protein known as JRAB on the collective migration of the cells. Okay, and we constructed two different mutants, delta CT and delta CC. So these are two different mutants, and we tried to determine the effect of these two mutants on the traction forces that appear during the collective cell migration. And in the case of this mutant called delta CT, we found that so this is a collective migration of epithelial cells. So while they are moving from the upper side to the bottom side, downward, they are, as you see here, generating wrinkles in the border between the cells and free space. So while they are moving, they are generating large forces that can be visualized by the appearance of these wrinkles. Okay, but unlike this mutant, so that was shown with this purple color. In the case of this mutant, shown this light blue, we found that while the same cell is moving, but with different mutant of the JRAP molecule, we found that there is almost no wrinkle that appears during the collective migration. But we found that they can actually more quickly move compared to the former one. So they can more quickly move while they are generating less magnitude of forces. Okay, so that's our finding, and so our system was found to be effective in detecting the effect of specific gene mutations on collective cell migration as well. Okay, so now let me move on to another topic in which we tried to use our system to for drug screening. Okay, so actually now I'm at Osaka University, but before I coming to Osaka University, I was at the pharmacy department at Nagoya Institute of Technology. So where I learned that in the pharmacy field, they needed to test a huge number of candidate molecules to find a good drug. Okay, so to do that, we have to increase the throughput, not only based on by the improvement in software, that can be possible by, for example, introducing a machine learning system. But also we have to somehow increase the throughput by, for example, by improving the hardware. So to do that, we initially used this type of conventional cell culture dish or Petri dish, but we manufactured cell culture plate, cell culture plate, like 24 or well plate or 96 cell culture plate. And by using this system, we can combine our cell culture plate together with high throughput oriented devices such as microchannel pipettes and specialized plate readers. So by combining all these together, and so this is the actual apparatus that we made, and this is the actual well plate 24 well plate, and by using this system, we determined. So we used drug library. So here's the list of the drugs that we tested, and by using our system, we determined how the effect of treating cells with these drugs result in upregulation or downregulation of the several forces. And we also simultaneously measured the extent of the migration. So this might be a little tiny in size, but what we did for this experiment is that we did a scratch assay or wound healing assay. So you may know that wound healing assay, but to determine the migratory potential, we scratch a part of cells within the cell colony, and by observing how fast the wound is going to be closed, then we can evaluate the extent of the migratory potential. So by doing these two experiments, we got a result like this. And if we focus on the change in the force, for example, for example, cytokeratin D is a well-known actin polymerization inhibitor, and lateral A is also another famous actin polymerization inhibitor, and jasprekinolidone is an inhibitor for actin depolymerization. So it is well known that by using these low molecular weight compound, it is known that actin-based structures are disrupted, which in turn decrease the level of the cellular forces. And maybe brevethatin and Y compound are also well known to inhibit the activity of myosin. So we found that these famous drugs result in decrease in the level of the force. So here, please note that force index of 1 means that there is no change. So here's there is a control here. So control is almost in the close to the this line. So it means that there's no change. But we focused on these two. So calcium A is actually another well-known low molecular weight compound that is known to increase the level of the force. So in the basic cell biology study, we have sometimes situations that we want to increase the level of the force to evaluate the effect of increasing the forces. Okay, but calcium is actually not a good drug, in my opinion, that I'm going to talk about later. But what we found was that JNK inhibitor 8 has a similar effect with calcium A, and so to my knowledge, to the best of my knowledge, JNK inhibitor 8 is not known to increase the magnitude of the force. So we focused on this chemical compound. So as I mentioned, by treating cells, so these are actually HeLa cells, cancer cells, and by treating these cells with calculin A, that is actually known to increase the level of the force, we often observe a rounding of the cells. So this is a percentage of how much cells are going to get round in shape, because by treating the cells with this drug, cells are detached from the substrate, and once they are detached, they never return to the original shape, or they never get spread. So it means that by treating the cell with this drug, cells will die. So actually, we want to it would be better to avoid using this chemical or compound, but because there is a limitation in choosing a good chemical or compound to evaluate the effect of increasing the level of the force. But our system found that JNK inhibitor 8 actually has a similar effect with calculin A is doing on the cells. But we found that unlike the case that cells are going to become round in shape after the treatment with calculin A, but we found that with this drug, it's really rare to observe such a rounding, cell rounding. So it's almost similar compared to vehicle control. So in terms of that, JNK inhibitor 8 may be a better inducer of contractile force compared to calculin A that has been typically used in the field, particularly in the field of mechanobiology. So you might think that by decreasing the concentration of the so actually these are typical concentrations that are used in the basic study to evaluate the effect of in this case. These are actually a serine threonine inhibitor, and JNK inhibitor is literally an inhibitor of the JNK activation. But you might think that by decreasing the concentration of calculin A, you might think that such a bad effect could be avoided. But we found that by decreasing the concentration down to 0.5 nanomolar or 0.1 nanomolar, we found that the force is no longer increased with this lower level of concentration. So we have to use this calculin with at least this concentration. But as long as we use this calculin A with this concentration, it results in detachment of the cells. So in that sense, it would be better to avoid the use of the calculin A. And we also did Western blotting to evaluate the phosphorylation of MRLC. This is myosin regulatory light chain. So to increase the cellular contractile force, no muscle myosin must be activated, and to do that, MRLC must be phosphorylated. So this is actually a typical biochemical marker, or maybe to my knowledge, this is the only biochemical marker to evaluate how the force is going to be increased. And so we did Western blotting, and we observed a tendency that by treating the cell with these inhibitors, we found there is that's the increase in the. So this is so actually MRLC can be phosphorylated at the two sites, and so these are double phosphorylation, and this is single phosphorylation. And in this case, double phosphorylation is a little lower compared to the case of calculin A, but for the single phosphorylation, we did observe increase in the level of the phosphorylation. So even with this chemical observation, we found that JNK inhibitor would cause an increase in the phosphorylation of the MLC, which in turn will increase the level of the force. Okay, so we also did microarray analysis, and we found that by using calculin A, because cells will be detached from the substrate, which will eventually induce some pro-inflammatory signaling. So by comparing the cells treated with calculin A or cells treated with JNK inhibitor, we examined how the gene expression is going to change, and we did observe a difference between them, and particularly we focused on because they are going to be detached, so that might finally induce apoptosis, and to induce the apoptosis, pro-inflammatory signaling would be activated. And indeed, based on this microarray analysis, we observed that there is indeed an increase in the activation of the pro-inflammatory signaling related pathway. So this is still an ongoing study, but so far we have found that calculin A may not be good in terms of the activation of pro-inflammatory signaling, which in turn will induce apoptosis of the cells. But unlike the calculin A, JNK inhibitor is more gentle, so it will not such bad effect on the cells. Okay, so actually this is the last slide to summarize my talk. So we have developed machine learning based wrinkle force microscopy, which allows us to do the cellular traction forces in high throughput manner. And using this technology, we have so far found that JNK inhibitor 8 might be a better inducer of cellular traction forces compared to calculin A. Although this one has been conventionally used, but because this will cause some pro-inflammatory signaling events, so this might not be used in terms of the toxicity that might be given to the cells. Okay, so finally, I'd like to acknowledge my collaborators, and as I mentioned in the middle of my talk, Hong Lee is my really good PhD student, and Dr. Matsunaga Daiki is actually one of the corresponding authors of the manuscript of the GAN system that we are now submitting to a journal, and Dr. Matsui also made a big contribution to this work. And these are persons who made a contribution to this work, and these are my funding sources. So that's all for my talk. So thank you very much for your attention.
Thanks a lot. It was fantastic, very, very refreshing and super interesting. So while the other warm up with the questions, I have a comment. Indeed, we also use caliculin A and nocodazole to induce tension in epithelial cells. And we often found, or basically we dropped caliculin A because it was very unreliable. So to confirm your data here. So yeah, it's very good to you. You have these under publication, so we can also cite it. Great. Okay, so are there any questions?
Yeah, I would have a question. Yeah. Okay, so when you show your wrinkle system, I wanted to ask you, what is the lower limit of detection of force? Because you showed this, I don't remember this cell line where you're knocking down a specific focal adhesion associated proteins, and you don't see any wrinkles in the monolayer, in the migrating one. So I'm just wondering, yeah, which one is the minimum detection limit that you have?
So actually, Actually, we still tested only specific cells. So I think that the threshold will be dependent on the types of the cells. But no, that depends on the stiffness of the substrate. And here we used a stiffness of 10 kilopascal. And by using the 10 kilopascal or substrate, or the minimum force required to generate forces are about 10 Pascal, or by, for example, by multiplying the area of the focal adhesion, that will make the actual force.
So yeah, now indeed, this is associated to my follow-up question that was indeed if you can change the elasticity of your substrate, I mean, of this fluid, silicon fluid. So yeah, you already answered the question. Thank you. Okay, then I see Mauricio. Mauricio, if you want to. Thank you, Shinji.
Thanks, Shinji, for the presentation. It was very nice, very clear. I do have a few questions. It was not clear to me after you trained the GAN network, you are able to retrieve the localization and the orientation of these vectors, but also the scale or not the scale of the vector. So in physical units, I mean.
Okay, so you are asking scalable what?
The scale of the force, let's say. You are able to obtain in newtons, let's say, or just the relative strength of the forces.
So we can get the absolute value of the force as a unit of Newton, but here we are showing Pascal, because typically in the case of conventional attraction force microscopy, Pascal rather than Newton is used. So that's why we showed with Pascal here.
Okay. Okay, thanks. And my next question is about the error. So I guess that after you train the network, there are some kind of errors, and those errors may be located in specific regions of the cell. So are any geometrical conditions, let's say corners, so just throwing ideas, where you have more errors than other areas?
Yes, indeed there is an error in our systems, but we are not still sure if there is a specific location where error can be easily induced. But yes, there is indeed an error, but at least I am not aware that there is such a specific location of where error can typically appear. So so far I'm not so far we are not sure the typical position of the.
Okay, and just last question. About the GAN network, you use like a standard GAN with a standard objective function, or you have to modify the objective function of the GAN network?
Actually, this was, I'm sorry, but actually this was almost done by these two guys. So actually, I may answer the question.
Okay, thanks.
Rosario, thank you, Mauricio.
Thanks. First of all, it was a really interesting presentation, so thank you. And I wanted to ask, is there is a certain point that the substrate has a plastic deformation? Like either over time?
No, I don't. This is totally reversible because this is totally elastic. So once you remove the cells, the rings become totally planar again. So there's no plastic deformation.
Okay. That's it. Thanks.
Okay, Sebastian.
Thank you very much for a very wonderful presentation. I have a very quick question. Have you used this system for studying bacterial biofilms, or do you think it could be used for this, or it could have a benefit to study some mechanical properties of biofilms?
Well, we haven't done on the bacteria, but so my answer is I don't know that yet, but I think the bacteria will produce a low level of force. So in that case, maybe we have to lower the stiffness of the substrate to allow the bacteria to induce wrinkles. So in that case, now we are using elastic modulus of 10 kilopascal. That's actually very low. So by further decreasing the level of the stiffness, it's like a fluid. So it would be very difficult to detect such a small level of the force. But that's very interesting point.
Thank you.
I would have another question, if I can.
Yeah, sure. Go ahead.
I was just wondering, Shinji, if you are thinking to upgrade your wrinkling algorithm to basically look into cells in 3D. So because now you are showing everything on one. Yeah. Okay. You got it.
You are a developmental biologist, so you are more. You might be more interested in 3D. Yes. Now. Because we have to use the wrinkle system, so our system is so far applicable only to 2D system. But the idea of using GAN will also be applicable to 3D tissue. So actually now we cannot use the wrinkle system, but we are now trying to get training data for the GAN system for 3D tissues using collagen system, and by getting a large number of data. And once we got a large number of data, enough to allow the GAN system to learn the relationship. So for example, by just observing a specific marker, fluorescent marker, and if the GAN system understands the way of converting from the specific fluorescent marker to the actual force field, then by just observing the fluorescent marker, that must be easier to
extract them.
So so it allows us to speculate the force fields that may be possible in the future.
Yeah, cool. Okay.
I also have a couple of questions. I was a good guest this time and let everybody speak before me. Usually I don't do that. But so the first one is related to the experiments where you show with the mutation that collective migration is faster at lower traction forces. How do you explain that? What is your hypothesis there?
So in the case, so let me show you the slide. So in the case of this mutant, they are generating larger forces, but they move slowly. So it's like while, for example, you are walking, but you are, for example, it's hard to explain, but they are loading a bigger force onto the substrate, but they move slowly. It's like they are trying to confirm the substrate, the stiffness. It's hard to say. Well, in the case of this one, so it more quickly move while exhibiting less magnitude of force. But I think that it is possible because it's like a fluid motion. So in the case of fluid, the force will be much smaller compared to the solid movement, but they can more quickly move. So similarly, in this case, they are exerting less level of forces, but that would be why they can more quickly migrate. Because the connection between the substrate and the cells are
So they have a lower friction that, so they're moving faster because they have lower friction and higher turnover of focal adhesion.
Yeah, I was going to tell you, probably the focal adhesion are less mature, so they don't form this giant mature adhesion, and so they don't really apply force, and it's more distributed probably, and so the level of detection, yeah.
Yeah, that's what I wanted to say.
That's great. That's great. And then I have also a methodological question, because I think that one of the limitations also related to what Mauricio was asking on the detection of these wrinkles, it is possibly due to the optics of the system. So because you do have a cell at the top and you're visualizing with phase contrast, then you see the wrinkles only where the cells is not. So my question is if you have tried to look at these wrinkles using reflection interference microscopy, so in reflection mode.
So we have so far tested phase contrast microscopy and DIC, and you mentioned that reflection interference. So I think that it will be possible because there is out of plane deformations in the substrates that will be visible even using RICM that you mentioned.
Right. Right. Because that was one of the techniques I was trying to develop when I was back in Singapore, but then I left and didn't have time to continue on that. I think it would be a nice combination because with the reflection interference, basically you then don't see the cell at the top. The light will be reflected by your interface, the first interface.
That's really interesting.
That's great. Okay, are there other questions?
Yes.
Nicholas?
I have a question related to, well, thank you for the talk. It was a great talk.
Nicholas, get closer to the microphone because we don't hear you.
I can't hear you very well.
If there is a connection of the wrinkles with fingerprints formation, if you know, on the time scales or on the spatial scales also of fingerprints formation.
So sorry, I mean not follow your point. So can you say what?
Yeah, if the wrinkles that you see in the substrates are anyhow related to the fingerprints.
What do you mean by single what single print?
Fingerprints.
Patterns.
Okay. I don't know much about fingerprint, but in the case of our wrinkles, so the substrate is originally planar, so it's totally flat. But once cells grab the surface to pull, then wrinkles appear. So, but in the case of maybe fingerprint, there is always wrinkles there. So I think there's.
I think it's a different length scale, Nicholas. So the wrinkles that they're looking at are in the nanometer to micrometer scale.
Okay, so can I show you there? It's actually depend on how much force you are applying, but
Shinji, you are not sharing at the moment. You stop sharing
already. So
yeah.
Let me do that again. Can you see that right?
Yes, now can.
Yeah.
So it depends on the cell types and depends on the location. But typically, for example, from the peak to bottom, this one has about 200 nanometer deformation. So this was obtained by simultaneously using phase contrast microscopy and AFM, atomic force microscopy. And so this is the geometry that we got. So based on this information, their length of scale is about 200 nanometers.
It's beautiful.
Okay. Thank you.
It's a beautiful demonstration. I do have one more question regarding this. So you do see most of the most of these wrinkles to be parallel. So I was wondering if there is some sort of persistence of the wave of the wrinkles. And if you would have some forces that instead are perpendicular to each other, how the system would deal with those.
So it's hard to say, but wrinkles tend to align in a specific direction, but once you apply a force to the direction of the actual length of the wrinkles, then higher or larger forces will induce the larger forces will determine the direction of the wrinkle. So if you apply higher forces, then wrinkle will appear in the direction perpendicular to it.
And you said that this this maximum wrinkle persistent wave is perpendicular to the stress fibers. Is that correct?
Yeah.
Oh, that's beautiful. That's beautiful. Great. Okay, thank you very much. Are there more questions for Shinji? If not, I do thank you very much for taking time to present with us. Would you mind staying a few more minutes, so to discuss with me and Christina a bit more? Okay.
Yeah, and actually also Rosario will join us. One of the students. Yeah.
Yes. Okay. Thank you, everybody. Bye-bye.
Thank you. Thank you, Shinji. Bye.